Hello, my name is Kumar Sharma. I'm the Chief of Nephrology at UT Health San Antonio. Today, we would like to present an interesting study on the kidney-brain axis that was recently published in JCI Insight. Dr. Afaf Saliba, who is the chief author and production of the work, will present the study. Afaf, go ahead.
Hi, my name is Afaf Saliba. This work represents the centerpiece of my PhD dissertation conducted in the lab of Dr. Kumar Sharma at the Center for Precision Medicine. I would like to acknowledge the contribution of all co-authors and collaborators which made this research possible. Today, I will present our study on the role of tryptophan metabolism and its association with systemic inflammation, neurotoxicity in kidney disease. Kidney disease is linked to neurological symptoms, but the molecular mechanisms remain unclear. We hypothesized that kidney dysfunctions alter systemic inflammation, leading to the accumulation of neurotoxic metabolites. To investigate this, we used a mouse model of rapid kidney failure that allows us to study kidney failure without the widespread of multiple organ failure, and we performed targeted bulk metabolomics and found that tryptophan metabolism was the top pathway that was skewed in this mouse model. We saw that in the plasma level, particularly, there was decrease in tryptophan level and serotonin levels and accumulation of quinolinic acid. Metabolic dysfunction in kidney disease is often accompanied by inflammation. Tryptophan metabolism dysregulation is strongly associated with inflammatory processes at multiple levels. So we explored inflammatory markers in the kidneys of the kidney failure mouse model and found increase in CXCL1 and interleukin 1 beta in the kidneys. And at the systemic level, we observed significant increase in neutrophil to lymphocyte ratio, interleukin 6, and SUPAR level, indicating systemic inflammation and local inflammation in the kidney. Knowing that quinolinic acid is a neurotoxin and knowing that many of the tryptophan metabolites can cross the blood-brain barrier, even if it was intact, we analyzed the brains of these mice and we found that they also have increase in inflammatory markers, and we found increase in tunnel positive cells showing apoptotic cells at the cortical areas. Moreover, multi-mass spectrometry imaging showed us a striking event where we see that quinolinic acid is accumulating specifically at the ependymal cells. These are glia cells that help regulate cerebrospinal fluid in the ventricles. In order to confirm that our results are not only part of the kidney failure mouse model that we used, we also tested the same pathway in a severe ischemia mouse model of 35 minutes ischemia sacrificed after 24 hours. You can see with plasma creatinine and BUN level how severe the acute kidney injury. And we saw the same pattern of decrease in tryptophan, accumulation of quinolinic acid in the plasma, and plasma kynurenine to tryptophan ratio indicating degradation, accelerated degradation of kynurenine pathway. This was correlating with plasma creatinine and BUN levels. Moreover, the brains of these ischemia-induced acute kidney injury mouse model also exhibited an increase in inflammatory markers. To determine whether these effects were specific only to acute kidney injury or to kidney dysfunction in general, we also tested the same pathway in an adenine-induced chronic kidney disease mouse model. And we found that plasma quinolinic acid was significantly and very highly correlating with markers of kidney dysfunction and markers of inflammation. And I'm not showing here, but also in the paper you can see plasma, not only plasma quinolinic acid, but also brain quinolinic acid was significantly increased, as well as kidney quinolinic acid was significantly increased and correlating with markers of kidney dysfunction and systemic inflammation. Interestingly, we also found with multi-mass spectrometry imaging, multimodal analysis, that quinolinic acid was specifically accumulating at regions of tubular damage in the chronic kidney disease section. We did not find any quinolinic acid in the controls. However, in the CKD model, we saw accumulation of quinolinic acid at the tubular dilation areas. To add a translational aspect to our study, we analyzed patient samples and found that with advancement of CKD stages, there was significant accumulation of quinolinic acid in the plasma, which was correlating with kidney dysfunction and as well was correlating with lower quality of life caused by fatigue. In brief, our study provides strong evidence that kidney dysfunction is associated with systemic inflammation and tryptophan metabolism dysregulation, leading to the accumulation of quinolinic acid in both kidney and brain. Future research will focus on understanding the mechanisms of quinolinic acid toxicity, developing interventions to mitigate its harmful effects by targeting the kynurenine pathway enzymes, ultimately improving quality of life for chronic kidney disease patients. Thank you for listening. You can read the paper at JCI Insight.
Thank you, Afaf, and congratulations on an excellent piece of work. Please contact Afaf or myself if you have any questions. Bye-bye.